Hello and welcome everyone to this podcast for human genetics. In this podcast, I'm going to continue our discussion of the cell. In particular, I'm going to talk about cell division and cell death. When we think about cell division, it's important to talk about the cell cycle. The cell cycle has two phases called G phases. The first is G1 and the second one is called G2. G stands for gap. The phase between the two gap phases is called S phase. And then the phase between G2 and G1 is called M. For our purposes, M can be mitosis, which we'll talk a little bit about today, or meiosis, which we'll talk about in a podcast or two from now. Okay, so as we begin this discussion, what I'd like to do is take a single cell through this cell cycle. And I'm just going to draw one pair of chromosomes. Now this cell will go through G1. As it's in G1, it will check to make sure it's the correct size, no mutations, and the enzymes ready for S phase are available. The cell will then go through S phase, and while it's in S phase, the chromosomes will duplicate. And I'm going to show the copied chromosome, the new copied one, as a dashed line just to show it appearing different. So S phase, what happens here is DNA is copied. S stands for synthesis. Now the end of S phase, what will happen is the DNA will completely have been copied. So here are the two original ones, and the copied ones I'll draw like so. During G2, the cell grows. The cell will check for mutations, and it'll make sure that enzymes for M phase are available. Collectively, and I'll draw this up here, G1, S, G2 are called interphase. So that would be all of this is interphase. Now what happens in M phase? We're not going to go through the gory details of the various components of mitosis. We'll do that for meiosis later, but for mitosis, well, all I want you to know is this, that these chromosomes will pull apart to produce two identical cells. So each of these identical cells will now have the exact same chromosomal content as the original cell. Now these cells can go on and copy again, can go and re-enter the cell cycle and keep growing. So it's really important to know that the purpose of mitosis, that is this cell cycle starting with this cell and ending up with two identical cells, is that mitosis, I'll draw it up here where I have a little bit of room, produces clones or identical copies of the original cell. For instance, in my pancreas, when cells divide in my pancreas, I want them to produce new pancreas cells. I want no variation from one cell to the next because I want them all to be able to do the same job. When variations start to occur, that's when things like cancer can start to set in. Now, we'll go over this in much more detail when we talk about cancer, but I do want to at least introduce to you the idea of cell cycle checkpoints. And we have one right here, we have one right here, and we have one right here. And so I'll make a little key down here. These are cell cycle checkpoints. All I want you to know about the cell cycle checkpoints for now is that Their job is to stop the cell cycle at these specific spots in order to maintain proper cellular division and cellular function. If they detect errors or a failure to separate the chromosomes in M phase, it will stop the cell cycle immediately, either fix those problems or kill the cell. Now, we will come back to these 3 cell cycle checkpoints later on when we talk about cancer, but that's all you need to know for now. Now I'd like to move on to something I just stated, but I would like to clarify a little bit more. And that is, why is it that some cells have a pathway in them that leads to their destruction, that is, to their own cell death? All right, so now let's spend a little bit more time talking about the cell death aspect here. Now, a cell could die just because it becomes damaged. You burn your arm, you cut your skin, some kind of structural damage occurs and the cell dies because of that. That's not what I want to talk about here. What I want to talk about here is something called apoptosis. So apoptosis is an orderly and rapid destruction of a cell. Essentially what happens, and I won't go into great detail, but you should know that this cell here will begin this apoptotic pathway by disforming. It will then begin to kind of chop itself up into these bags of the leftover cell. Then what happens is our immune system contains these white blood cells called phagocytes. And they will engulf all of these parts of the cell that just broke apart here and digest them. So they'll bring them in, and then you'll end up with a phagocyte that looks something like this, that just brought in all of this old dead cell, and it will digest those. Now what happens during this process, again, I don't need you to know all the details of this, but this is a cellular event. So I do want you to know this, that as the cell is misforming as shown here and begins to break down, this is all because of various organelles breaking down. The nucleus breaks down and the chromosomes inside do. The mitochondria, I'm just going to write mito here for short, and the cytoskeleton. And I should also say the plasma membrane. I'm just going to put PM for plasma membrane. These all break down. In doing so, they lose their ability to communicate with healthy cells, and they attract the white blood cells to destroy them. Now, I said something earlier, and you may or may not have caught it, but I said that most cells undergo cell death. However, not all cells go under this programmed cell death. We have some cells in our body that we call stem cells. Now, they can, of course, die, but in general terms, we think of stem cells as being immortal. They don't readily undergo apoptosis. We need to maintain a constant supply of stem cells. Now, I want to talk about the general function of stem cells, looking at an adult organism first, and then we'll talk about the different kinds of stem cells. And to begin this discussion, what I'd like to do is draw this liver. The liver has many important functions. It helps in detoxification of toxins when they enter our system. They help store molecules that we need. They do a lot of things, but I'm not going to ask you on the test what a function of a liver is. But what you should know is that cells that make up the liver will at some various points undergo apoptosis. And they will enter this programmed cell death and they will die. We know that every cell has a finite life. That is, except for stem cells. So within this liver tissue, we will have stem cells. What these stem cells will do is they can replace these cells that have died. And so I'm going to redraw this stem cell. I'm going to draw it over here. We know stem cells, when they divide, they will produce 2 cells. One of these cells will go on and differentiate to these specialized liver cells. The other recycles and maintains its stem cell status. So every time that stem cell divides, or most of the times when it divides, it will make one cell that differentiates and one cell that remains a stem cell. Most of our tissues in our body have stem cells, or what we often call adult stem cells. These are important in order to repair tissue that's damaged or to replace cells that have undergone apoptosis. A term your book uses, which is probably a good term to think about when we think about remaining a stem cell, we call that self-renewal. Now what I'd like to do is to talk about 3 types of stem cells. The first will be embryonic stem cells. The second will be somatic stem cells. Now, this is the type of stem cell that in the popular media we often hear called adult stem cells. I don't like the term adult stem cells to describe these, and I'll explain why in just a moment. And the third type is something we call induced pluripotent stem cells. We often just refer to these as iPS cells. Okay, so let's spend some time and talk about the embryonic stem cells. To do that, I want to give a brief background on early embryonic development. We'll go into more detail in this a little bit later, but for now, know that this fertilized egg here, after a sperm fertilizes an egg, is called a zygote. It will then undergo several cell divisions. At some point, say between days 4 and 5, an embryonic stage is reached. I'm going to draw it like this. Essentially, you have a whole bunch of cells surrounding this mass of cells. And it's this mass of cells I want to talk about. This is called the inner cell mass. All right, let me tell you what this stage is called. It's called the blastocyst stage. Now, the blastocyst will continue embryonic development, where it will form the very structures of the embryo, the organs, the tissues, all those parts. And then it'll enter the fetal development stage, and ultimately, if all goes as planned, the baby is born. Now, the reason I wanted to focus on the blastocyst was because of this inner cell mass which forms in this stage. This inner cell mass, that is our source of embryonic stem cells. These cells in this inner cell mass are something we call pluripotent. They can become any specialized cell found in any tissue of the body. Pluri for multiple, and then potent. They have the potential to form any one of these cells as development continues. So they have a lot of potential. So in the media and in the literatures, when we hear the term embryonic stem cells, it is these cells that we're talking about. All right, let's talk about adult stem cells, or what I prefer, somatic stem cells. Somatic stem cells are something we would call multipotent. They don't have the same potential as an embryonic stem cell. They can't become any body cell. But a multipotent stem cell, that is the somatic stem cell, has the ability to become certain types of cells. So for instance, a liver stem cell has the potential to become one of many types of liver cells. A pancreatic stem cell can become one type of many pancreas cells. However, a stem cell found in my liver could not readily form a bone cell, for instance. It still has a lot of potential, but not quite the same as an embryonic stem cell. Now, the reason I don't like the term adult stem cell is because these stem cells are found, of course, in adults, as you would expect, but they're also found in embryos. If they're found in a fetus, they're found in the umbilical cord. Because remember, once they leave this blastocyst stage and they start forming specialized structures, they're no longer pluripotent. So in the early embryo, when some of these cells specialize and become pancreatic stem cells, they've lost some of their potential. So again, adult, embryo, and a fetus, and umbilical cord, all of these contain somatic stem cells. Next, I want to talk about induced pluripotent stem cells. Next, I'd like to talk about induced pluripotent stem cells. So first of all, these iPS cells are not naturally occurring. These are stem cells that we produce in the lab. So what can be done is, I'm just going to draw a stick figure here. We can take a single cell from this individual here. So here's that cell from that individual, and we can reprogram it. This somatic cell to behave like a pluripotent stem cell. So remember, a type of pluripotent stem cell is an embryonic stem cell. And all we've done here is take any old body cell and have changed the way genes are expressed in it to make it think, so to speak, that it's a stem cell. And what I think is pretty interesting is that of the 23 or so thousand genes we have in our cells, all we have to do is overexpress, indicated by an up arrow, 3 genes. Increasing the expression of these 3 genes makes a somatic cell, a body cell, behave like an embryonic stem cell. Some of the advantages of these iPS cells is that researchers can use them in such a way, researchers can use these similarly as they would use embryonic stem cells. All right, the last thing I want to talk about in this podcast is the human microbiome. This turns out to be a growing field of interest, and we are discovering that the human microbiome is incredibly important to maintaining human health and can be the cause of some human diseases. So what is the human microbiome? If we look at one of my famously drawn human beings here, we know that this human contains trillions of cells. However, the most abundant cell in humans is not a human cell. Sounds confusing, right? That is because our most abundant cell are bacteria. So bacteria are the most abundant cell in humans. So the human microbiome are all the microbes that live in or on us humans. At first, when you think about it, it seems a little creepy, but these microbes allow us to survive. And all they ask of us is to give them some food every now and then. Seems like a pretty good deal. But what I'd like to do now is talk about 3 or 4 examples of why the microbiome is important. Some skin bacteria keep our skin clean and prevent things like acne. We've known that people who have weight loss surgery have a lower blood sugar compared to before the surgery. And this has nothing to do with a change in how much they're eating or what they're eating. It actually turns out this is in part due to a change in gut bacteria. That surgery changes the overall dynamics of the bacteria in our gut that leads to a lower blood sugar. In malnourished children, a change in a microbiome quickens starvation. There has also been some links between the human microbiome and various conditions like ADHD, autism, and certain behaviors. So to understand the human body, often we're discovering that we also have to understand the microbes that live within our body. Well, okay then, that ends this podcast where we talked about cells, cell division, apoptosis, stem cells, and the microbiome. If you have any questions at all, please let me know or talk to members of your team, and I will talk to you later. Bye.